Hello everyone, today I'll be teaching you about PCR and ELISA testing. Now, first off, what is PCR? PCR stands for polymerase chain reaction. PCR is a process that involves copying short pieces of DNA in order to make millions of copies in a short amount of time. This can be useful so that doctors can see if someone has a particular gene that's making them sick or if a certain virus is present. And it should be noted that a thermocycler is used so scientists can place the exact time limits on when the temperature should change. So for those of you who don't know, a thermocycler allows the scientists, biologists, whomever is using it to input basically a certain temperature they would like, and then how long that temperature should be, and then how quickly it should change to either a warmer or lower temperature. Now, the steps of PCR. So first, a piece of single-stranded DNA is placed into a thermocycler. Next, denaturization occurs when the temperature lowers. This is in order for the primers to attach. Then the temperature will raise slightly. Next, annealing occurs when the two strands that begin and end with the primer start to appear. Soon after this occurs, 8 fragments are produced during the process of extension and elongation. Then steps 2 to 4 will be repeated until there are millions of copies, and then these are processed by biologists, doctors, scientists, whomever happens to be working with the PCR. Now, our second test is ELISA. So what does ELISA stand for? It is an enzyme-linked immunosorbent assay. So ELISA is an in vitro test that can be used with either antibodies or antigens. Then they are exposed to a complex of an enzyme that is linked to the antibody or antigen. So if you're using the test with antibodies, it would be exposed to the complex of an enzyme linked with antigens and vice versa. Or an enzyme linked to an anti-immunoglobulin specific to the antibody. This is followed by a reaction of the enzyme with a substrate, which shows a color change corresponding to the concentration of test material. Now, I know this sounds really complicated, but hopefully when I show the steps, it'll make more sense. And a quicker way to remember this, so essentially either an antibody or antigen is chosen, and they're exposed to a complex of an enzyme or an anti-immunoglobulin with the opposite of what they are. So antibodies would be exposed to antigens and vice versa. Then this is followed by a reaction of the enzyme with the substrate. And the substrate is used to show a color change. And the color change shows whether or not they would have the virus. For instance, if the color change is shown, like if the test tube turns blue or whatever, they would have the virus. But if nothing changes, if there's no visible color change in the test material, then they probably don't have the virus. So the steps of ELISA. First, an experimental sample is placed into the well, and next the proteins will stick to the sides of the well. Then a detergent is added, which washes out the unbound protein blocks that remain on the surface of the wells. Then a primary antibody binds to a specific antigen. This is very similar to the proteins that bind, except this time it's the primary antibodies. Then a similar thing happens where the unbound primary antibody is washed away. Then this process somewhat repeats itself, except this time it's a secondary antibody, and the secondary antibody has the enzyme-linked colorimetric tag attached, which sounds really complicated, but essentially this enzyme is what's going to make it change color if there is a presence of the antigen in this case. Anyways, this is added to the well and it binds with the primary antibody. Then the excess secondary antibodies are all washed away. Then the substrate is added to the wells. The substrate will begin to bind to the secondary antibody, and this is what will cause the color change if the antigen is present. If the antigen is not present, there will be no visible color change. Congrats! Now you better understand the steps of PCR and ELISA testing.